Okay, well, you started. Okay, so you started the recording. Okay, I'll just give it maybe 1 or 2 more minutes. I just sent a reminder to Vid. So this is my pleasure to introduce the CFAR seminar today. Unfortunately, the main speaker, Dr. Bergert, canceled because, yeah, he got COVID, so he's not feeling so well. But we have a new face seminar speaker today, Dr. Shashidhar Ravishankar. Before I introduce Shashi, I have some housekeeping. So as you all know, the CFAR seminar provides forum for presenting and discussing state-of-the-art research findings in HIV pathogenesis, virology, care and treatment, and to provide an opportunity for ongoing enhanced communication and multidisciplinary collaboration among HIV/AIDS researchers across the 5 CFAR affiliated institutions and their partner organization. Just to let you know that the next CFAR seminar, and I think will be the end for the year 2021, will be June 3rd. It would be Nicole Doria-Rose from Chief of the Humoral Immunology Core from the Vaccine Research Center at NIH, and she will be talking about an evolutionary arms race HIV-1 versus broadly neutralizing antibodies. So now today, new faces is Shashi, and he's a bioinformatic analyst in the Warren Lab at Fred Hutch. He attended Georgia Institute of Technology, where he got his PhD in bioinformatics. His dissertation research was conducted in collaboration with the malaria branch at the Centers of Disease Control and Prevention, with a focus in developing scalable and reproducible frameworks for genetic epidemiology of malaria. At the Warren Lab, his focus has been on developing bioinformatic frameworks and methodology to analyze high-dimensional data generated from single-cell sequencing and immune repertoire sequencing. So happy to have you, and forward to your talk. Looking forward to your talk. Thanks.
Thanks, Maria. Good morning, everyone. Today, I wanted to present our work on the effect of long-term ART on the repertoire diversity in individuals living with HIV. Before I proceed, I just wanted to disclose that I have no financial disclosures or conflict of interest with the presented material in this presentation. To start off, I wanted to acknowledge what a massive effort the study that the UW Fred Hutch CFAR trial has enabled here in gathering such a large repository of data and collating all the information. And I and everyone at our lab is really grateful for this collaboration. I just wanted to highlight to everyone here that despite the prevalence of a lot of therapeutics for HIV, the burden of HIV still remains global, with a concentration of this in the sub-Saharan Africa region. In 2019, about 38 million people were known to be living with HIV. Around 1.7 newly infected people were diagnosed with HIV, and nearly 700,000 deaths were recorded in HIV-related, to be HIV-related. And it becomes important for us to still continue, though there's been a lot of work on understanding the effects of HIV and ART on the immune repertoire, we still need to understand long-term modifications in the immune repertoire with the start of ART. There has been some research at looking into long-term effects of ART. I just wanted to quickly highlight a few papers. A study published by Anthony Fauci's lab looked at CD4 counts, CD8 T cell counts, and B cell counts in individuals living with HIV over the course of 12 months where they received ART, and they found that there is a negative correlation between the CD4 count recovery due to ART and CD8 counts. Another study by Yi-Kun Kwang's lab looked at the CD4 CD8 ratios in individuals living with HIV over the course of ART treatment over 8 years. And they found that individuals who showed immunological recovery through ART treatment had a increased rate of growth in the CD4 CD8 ratio. And this rate stabilized over 2 years of ART treatment. And finally, I wanted to highlight a study done by Benjamin Chain's group looking at the T-cell receptor repertoire in chronic HIV infections. They tracked the repertoire at 2 time points for individuals living with HIV, one before the start of ART and another 3 months after the start of ART. And they noticed that in short-term tracking of the effects of ART, they did not find much diversity in both, much increase in diversity in both the alpha and beta chain T-cell receptor sequences, whereas, they did not notice the repertoire reconstituting to the levels of previous individuals who were previously undiagnosed with HIV. Given that there have been fewer studies that looked at the T-cell receptor repertoire over a long-term ART, we wanted to explore the effect of ART on reconstitution of the T-cell repertoire in individuals living with HIV. To do this, we sampled 30 individuals from the CFAR cohort who were living with HIV. We were able to collect viral loads, CD4 counts, comorbidities, co-infections, absolute lymphocyte counts, and HLA typing from all these individuals, ranging over a time span of 2 to 29 years through the course of their treatment. We were also able to collect 198 unsorted PBMC samples on which we performed T-cell repertoire sequencing. And this amounted to about an average of 6 repertoires per individual over a time span of 5 years. To our knowledge, this is the largest characterization of the dynamics of T-cell repertoires in individuals living with HIV. We wanted to highlight that we made a deliberate choice here to use unsorted PBMCs, primarily because of the amount of PBMC samples we were able to recover from these individuals. And we prioritized the depth of sequencing for the study over resolution at the CD4, CD8 level. To start with, we collected unsorted PBMCs from pre and post ART specimens from the CFAR biorepository. We isolated genomic DNA from the PBMC samples, prepared NGS libraries, and performed QC using the adaptive immunoSEQ platform, and sequenced the T-cell beta chain T-cell receptor repertoire of all 30 individuals. This high-dimensional data set that we collected enabled us not only to look at CD4 counts over the course of treatment, but also identify specificity groups between repertoires which would confer antigenic specificity, look at TCR diversity using rarefaction curves, understand the clonal structure of the repertoire using rank abundance analysis, track the top frequency clones across an individual's repertoire with alluvial plots, and finally, understand the overlap between the repertoires over the time course of treatment using percent similarity index. To start off with, we looked at the viral load variation in these individuals with the initiation of ART, and found that in 28 of the 30 individuals from this cohort, initiation of ART caused a drastic reduction in the viral load, which persisted through treatment. Most individuals, the 28 individuals, showed a median viral load after initiation of ART of less than 200 copies per ml. Oh, I'm sorry. Are you able to see my slide transitions?
No.
Josh, your slides are not advancing. Click once into the window and then you should be able to click into the viewing window and then use the arrow keys on your keyboard.
Yeah. Let me try this again.
Can you go back one? Sorry. Slides, yeah.
Are you able to see the transitions now?
No.
I'm so sorry. Let me try to reshare the screen.
Can you go back or?
Yeah, I am moving back through my slides.
Can you stop and then start it again, the presentation?
Yeah.
Oh, yeah. So right now we're not seeing it.
Yeah, I'm just resharing my screen. Can you see the screen now?
It's coming up. No, I don't know. It's coming up, but nothing is coming up. So it's not, it just say that you start sharing your screen, but I don't see anything. Oh, here it is. Here it is.
Okay. Are you on slide? Can you see slide 3?
Yes.
Okay, great. I'll just quickly go to the slide I was referring to. But if you have any questions regarding earlier slides, you can come back to it at the end of the talk. I apologize for not checking ahead of time whether the slide transitions are visible. But
okay, so this is where you were. Just like make sure it's moving forward.
Yeah, I was at slide 10 trying to describe the viral load variations we observed in the cohort. So continuing from here, I just want to emphasize that with 28 of the 30 individuals, we noticed that after the initiation of ART, the viral load dropped, the median viral load dropped below 200 copies per ml, and we group these individuals as showing durable viral suppression through the course of treatment. In 2 of the individuals, we found that the viral load was the median viral load after ART initiation was greater than 200 copies per ml, and we classified these 2 groups as showing a lack of viral suppression through the course of ART. Looking at the CD4 counts varying within these 2 groups, we found that in the group which showed durable viral suppression, there was a significant recovery of the CD4 count through the course of treatment, whereas in the group that showed a lack of viral suppression, the CD4 counts then decay over time. Coming to absolute lymphocyte counts, we found that they remained fairly stable in the group that showed durable viral suppression. We suspect this is due to the initial expansion of CD8 compartment of the T-cell repertoire at the start of the infection, which kind of diminishes over the course of ART treatment and the recovery of CD4 counts. When we look at the same count information, but split it up over the years of treatment, we find that in the group that showed a durable viral suppression, the CD4 counts tend to increase over time and looked like they reach a plateau around 2 years into the treatment, whereas the group that lacked viral suppression had a decay of the CD4 count over the course of treatment. We observed that the absolute lymphocyte counts remain fairly stable for the group showing a durable viral suppression, whereas the other group showed a decay of absolute lymphocyte counts as well. The advantages of using T-cell receptor sequencing, however, is that it enables us to also look into a different dimension of the repertoire with our analysis. One way to understand the T-cell repertoire is trying to find antigenic specificity groups from the CDR3 sequences of these beta chain repertoires. Work done by Mark Davis's group at Stanford developed a tool called Glyph, which looked at sequence similarity between T-cell repertoire receptor sequences and identified patterns which would enable, which would confer a similar antigenic specificity between these sequences. David Coffey from our lab developed a tool called T-cell pack, which allows us to visualize the T-cell repertoire in a comprehensive manner. Here, the larger, lighter circles represent the specificity group determined by Glyph. The smaller, darker circles represent a T-cell clone. This analysis this visualization allows us to look at the overall diversity of the T-cell repertoire, as well as diversity at the predicted antigenic specificity level as well. To quantify this diversity, we used Gini coefficients. The Gini coefficient is calculated based on the Lorenz-Curve distributions. It measures the equality or inequality of frequency distributions in a repertoire. It is calculated as the area within the Lorenz curve divided by the total area of this triangle here. And it ranges between 0 and 1. 0 would indicate total equality or diversity in the repertoire along the line of equality here. And a value of 1 would indicate a high degree of clonality in the repertoire. Here I demonstrate this concept with 3 different T-cell repertoire Lorenz curves. As the area under the Lorenz curve, within the Lorenz curve increases, we find that the repertoire moves from more diverse to more clonal in nature. We can use this Gini coefficient calculation to look at the global repertoire Gini coefficient, that is considering all the T-cell frequencies from the repertoire and calculating the Gini coefficient of the entire repertoire, or we can zoom in on these computationally predicted antigenic specificity groups and calculate diversity within these specificity groups, average them over all specificity groups identified in the repertoire to give the mean specificity cluster Gini coefficient. These 2 metrics measure different aspects of repertoire diversity. The first would give, as I mentioned earlier, the overall repertoire diversity, whereas the second would measure what is the extent of diversity within computationally predicted antigenic specificity groups. When we look at these 2 metrics across the 2 groups that we defined earlier, we find that in individuals who showed a durable viral suppression, the diversity over time improved at both the repertoire level as well as the antigen specificity level as well. One feature to keep in mind when dealing with T-cell repertoire sequencing is the variability in sampling that comes with sequencing. To normalize the depth and allow the comparison between different repertoires, we can use rarefaction analysis. Here we have the total number of unique productive T-cell sequences, receptor sequences on the y-axis, and the total number of T-cell sequences sequenced on the x-axis. The Rarefaction curve for one repertoire is generated by serially subsampling starting at 0, going up all the way to the actual sequencing depth represented by the red triangle here, and extrapolating this curve to an idealized depth. If we were to say extrapolate to 1 million sequences, we find that in the 7 repertoires that I have shown, the 4 at the bottom tend to saturate over 1 million sequences, indicating a lower diversity of T-cell repertoire, whereas the later lines have a higher slope, indicating an increased diversity over the course of sequence at greater sequencing depth. Another way to look at normalizing the T-cell repertoire counts is to subsample this data down to the lowest sequencing depth. This gives us depth normalized or rarefied counts. When we compared these depth normalized unique productive sequences prevalent in the 2 groups, we again found that with durable viral suppression, the total number of unique productive sequences significantly improved with treatment as compared to the group that lacked viral suppression. We tried to assess clonal composition T-cell repertoire data using rank abundance curves, where we had the cumulative frequency, the relative abundance of the sequence on the y-axis here, and the rank on the x-axis. And we found that the way to interpret a rank abundance curve would be if there's a gradual slope in the rank abundance curve, it would indicate equality in the distribution. A steeper slope would indicate inequality or oligoclonality in the cases of T-cell repertoires. While we looked at overall cohort information, a cohort level analysis up till this point, we wanted to highlight the ability to look at dynamics at an individual repertoire level when dealing with T-cell receptor sequencing data. So we are going to look, going further, we're going to look at 3 representative samples from the 2 groups we defined earlier. In the group which showed durable viral suppression, the median viral load, with a median viral load of less than 200 copies per ml, we found that with treatment, we observed 2 profiles. In one group, there was a consistent viral suppression in that as with the initiation of ART, there was a drop in viral load, which remained consistent through the course of treatment. Whereas in individuals who showed a variable level of viral suppression, there were blips or spikes in the viral load along the course of treatment, but overall, the median viral load was ended up being below 200 copies per ml. The third representative sample was from the group that lacked viral suppression. When we look at the diversity and clonal structure of these 3 groups using rank abundance plots and rarefaction curves and plotting out the number of unique productive sequences over the course of treatment, we see that they, at the time of ART, indicated the relative date corresponding to ART is indicated on the x-axis here. Around the time of ART initiation, the repertoire contained a very few unique productive sequences, but through the course of treatment, this gradually increased, showing the degree of reconstitution over time, or degree of, Looking at rarefaction curves for this individual, we find that earlier time points colored here in hues of purple showed a higher level of saturation in terms of their diversity. They seem to show a low diversity across, even when we extrapolate the rarefaction curves out to 1 million sequences, whereas later time points showed a higher slope, indicating greater diversity. Looking at rank abundance curves, we see that the slope of the rank abundance curves gets more gradual as treatment progresses, and the dominance of all oligoclonal or the oligoclonal structure of the repertoire tends to decrease with time, though there still seem to be large clones present at some later time points. In individuals who showed a lack of viral suppression, the unique productive sequences varied with treatment and never really reached the levels of those who showed consistent viral suppression. Looking at the rarefaction curves, we see that at all time points, the repertoire reached a degree of saturation fairly quickly. The diversity is never recovering with treatment. And finally, looking at the rank abundance plots, we can see that large clones persisted across treatment. And though there was a improvement at later time points with the rank abundance curve, there were multiple large clones which persisted through the course of treatment. And finally, looking at the group that showed variable viral suppression, we observe again that the number of unique productive sequences in these individuals varied across the course of treatment, but looking at the scales on the axes here, we can see that it was eventually comparable to the group that obtained consistent viral suppression. The rarefaction curves again show the same detail that at later time points, the curve has a greater slope comparable with the group which showed consistent viral suppression, indicating a recovery of the repertoire diversity with treatment. The rank abundance plots show that as treatment progressed, the slopes got more gradual, but there was a dominance of a single clone through the course of treatment. We can track these top clones across different time points within the same individual using alluvial plots. Here we show the top 50 sequences in an individual and track them across the course of treatment through the different repertoires we were able to collect from this individual. The red line here indicates the initiation of ART. Sequences are colored in purple if they were found in repertoires only before the start of ART, and colored in hues of orange if they were found only after the initiation of ART. Here we see that with the reduction of viral load at the start of ART, the clonality of the repertoire starts increasing, and we see the effect of these large clones diminish through the course of treatment. In individuals who showed a lack of viral suppression, we see that large dominant clones persisted through the course of treatment. The CD4 counts decayed with the start of, even after the start of ART, and viral load never really was brought into check. In individuals who showed variable viral suppression, we see that the repertoire dynamics of the top clones mirrored to some extent the viral load dynamics here. But overall, as treatment progressed, the effect of these last clones tend to diminish and as CD4 counts recovered. We then looked at compositional similarity between these individuals. We found that in individuals who showed a consistent viral suppression, the repertoire similarity was greatest at the time of ART, and distant as the repertoire is diversified with effective ART, the difference between the similarity between these repertoires also decreased, as indicated by the low similarity scores or low overlaps. If we look at the Tcell fact plots for these individuals, at the start of treatment, we see that the repertoire structure was very similar to the initial time points before ART initiation, but the furthest time point available for this individual, we find there's an increased diversity. The large clone seems to have shrunken in their effect in terms of the dominance in the repertoire, and the overall diversity has also increased with treatment. In individuals who showed a lack of viral suppression, the similarity or the overlap between the repertoires at different time points is fairly high until fairly into treatment. And we observe the same with the T-cell pack plots here, where large clones dominate the repertoire across all time points, even out to the furthest time point from the start of ART. In the group that showed variable viral suppression, we found that repertoire similarity, though it showed there is greater and overlap seems to be greater across the different time points collected from this individual, the final diversity of the repertoire compared to the initial time points is fairly higher. This is again reflected in the T-cell pack plots, where we see that as treatment progresses, the diversity of the repertoire also increases, but we do notice that there are a couple of large clones which persist across treatment. To summarize, we performed a longitudinal survey over a course of 29 years, where we collected the peripheral blood T-cell repertoire from 30 individuals living with HIV and tried to understand the effect of ART on the T-cell repertoire. We found that with durable viral suppression after ART, there was an increase in the CD4 counts. The absolute lymphocyte counts, however, remained approximately stable. Again, we suspect that this is due to the initial expansion of the CD8 compartment, which slowly regresses with the course of treatment. We found that the overall diversity of the T-cell repertoire increases with the course of treatment, and the diversity of T-cell clusters predicted using computational tools also seem to increase over the course of treatment. There was a regression of the oligoclonality in the repertoire and a shift towards polyclonality. And the composition of the repertoires evolved over time, where we noticed that the overlap between initial and later time points decreased as treatment progressed. With that, I'd like to thank the members of the Warren Lab, in particular Hudi Warren, Andrea Tarleton, Camille Peronin, and David Coffey for all their help and guidance on this study. I'd also like to thank members of the Schiffer Lab, Daniel Reeves, Joshua Schiffer, and Elizabeth Duke for their key insights in interpreting the data from the study, as well as members of the CFAR community, Balisa Mashadi Hussain, Dr. Nina Kim, Michelina Montano, Jacob Keen Candib, and the participants and their families for giving us an opportunity to work with such a rich data set. I'd also like to thank everyone who attended this talk today, and I will stop for questions. Thank you.
Thank you very much, Shasha and Shashi, sorry. And I'm glad you figured out the issue with the slide. So I see that there were some questions, so I'm going to go through them. I'm not a T-cell person, so I don't have any really question myself. But so first questions from Dev. Well, it was So I think it's from David Cole from the University of Washington. So it's more like comments and question. So I remember all literature about thymus atrophy in HIV. Do you know if thymus size recovers with ART?
That's a good question. I don't think we had the information for this cohort. I don't think we didn't have that information available for this cohort, but I am not aware. I'm not sure of the existing literature, and I'll review and get back to you on that. I'm sorry.
Okay. So then there is a literature that lymph nodes are permanently damaged. Is blown out thymus the reason why reconstitution is crappy? Do you know?
Sorry, I couldn't. I was just trying to go through the question myself. Could you repeat the question?
Oh, so is blown out thymus the reason why reconstitution is crappy?
In the individuals who did show a lower degree of our CD4 reconstitution, we cannot be certain whether it was due to their adherence, their lack of adherence to treatment, or was it any physiological characteristics within these individuals. That information was unfortunately not available to us. But in discussion with the CFAR lab, we concluded that it might be that the lack of reconstitution was primarily due to adherence issues to treatment.
Okay. So next, and I guess I don't think I can have people ask their own question. That would be probably easier. But so there is, as a clinician, I know that some people get poor, well, I think it's some, I'm not sure, get poor CD4 reconstitution as measured by simple PBMC assay, while others get great reconstitution by the simple-minded assay. First, why this variability? And second, is there a correlation between poor CD4 count reconstitution and poor CD4 repertoire diversification and poor CD8 diversification?
Yeah, that's a great question. And I think from our data, we do see that in those two individuals where we saw poor reconstitution, the repertoire diversity was fairly limited. There seems to be a correlation, but there are a lot of confounders here in the fact that we don't know whether they were whether their adherence played any role in the effect that we saw. But looking at the group that did achieve consistent viral suppression, we do observe greater reconstitution of the CD4 count, which is correlated with, fairly correlated with the repertoire diversification. So, and unfortunately, we couldn't distinguish between the CD4, CD8 compartments in this study, but looking at data of the total lymphocyte count and CD4 counts, we can see that there is all the individuals who showed an initial, a durable viral suppression, the CD4 compartment must have expanded initially and contracted over the course of time. So yes, a short answer would be, I think there might be a correlation between the CD4 counts and repertoire diversification.
Okay, so you have a lot more questions. I guess since we don't have another talk, we can go through them. I'm not sure, if people can unmute themselves, I think they can ask the question. I'm not sure if they can, but if you can, please unmute yourself and please ask Shashi the question. And if not, I'm gonna continue.
In a webinar, the people can't unmute themselves, but
they cannot.
If
okay,
you would like to select somebody that can be unmuted by me, I can do that.
Well, okay. Okay, I can follow up. So, so David Cole, and one last question. Have you compared, so GLI PH2 to TCR DIS3? And I'm sorry, because I don't know where they are. So.
Yeah, we started looking at CLIF2 and the advantages of that. This study was done with the older version of CLIF, and we plan to look at how different these two tools are, or what is the degree of similarity between the first version and second before switching over. So we are looking into both those tools.
Okay. So then Nuri Corey had some question. When you perturb the system, like with an influenza vaccine or VZV vaccine, can you pick this up?
Well, if we look at the amount of information we have on the repertoire, while it is the largest, there are gaps between the time points that we have detected. But if I can go back a couple of slides, if we look at, sorry, are you seeing these alluvial plots?
Yes.
Sorry again. Yeah, if you look at these alluvial plots, there is a slight correlation between changes in the repertoire with clinical events that we were able to capture for these individuals. Like in this final individual who was diagnosed with anal cancer and cervical cancer, we see some changes in the repertoire structure. It's hard to tease it apart, but we did observe quite a few public T-cell sequences from published literature. Here, if I can go back, the sequences colored in yellow or darker shades of purple are in fact public T-cell sequences that we identified. We weren't able to correlate it with any treatment that they received, if that answers the question.
And I think it was another one was how much of this repertoire dominance is due to HIV devoted responses or due to higher replication of endogenous flora like CMV or an EBV?
That's a great question. Again, we did notice quite a few CMV, EBV public T-cell sequences. So there can be some effect to that. We unfortunately were not able to tease it apart with this repertoire. Though looking at correlations between the viral load and the expanded clones that we could track across time points, we think there is a lot of this is governed by the HIV specific changes to the repertoire with the course of ART.
Okay, so I think there are a few more people who would like to ask questions, and now they are allowed to talk. So I think.
So I've unmuted. I have Ruann with microphone privileges, Florian, David. If anybody else would like to ask a question live, just contact me in the chat, and I'll let you turn on your microphone.
Yeah, so go ahead. Ask your questions.
Yeah, so this is Florian. Thank you so much for this talk. Very, very interesting. And I think actually kind of my question was already answered with your last comment or your last reply. Because during your talk, you didn't really say anything about the antigen specificity groups or the antigen specificities,
right,
and how they are changing over time with ART.
Right, so we looked at both public T-cell receptor sequences with non-specificity, as well as using computational tools to observe group our clones into antigen specificity groups. There were quite a few public T-cell receptor sequences that we observed across all the 30 individuals. They did not dominate the repertoire as much. They did show up, as you can see here, in the top 50 clones that we could track, but there were much more prevalent clones in this repertoire. So we kind of focused on computationally predicted antigenic specificity groups. We couldn't test the actual specificity of those predicted sequences, however.
Thanks.
Ruan, do you want to ask the question or?
Yes. Sashi, really excellent work and a very clear talk. Congratulations.
Thank you.
Can you speculate a little bit about the clinical implications for increasing polyclonality in the T-cell repertoire?
Yeah. Okay. Looking at just the data, and please, anyone can correct me. I think that what we find is, at least my hypothesis here is, with increased diversity or polyclonality, there is a more, the targeted response to any HIV in specific could be improved in the sense that the polyclonal nature within the antigenic specificity groups that we observe to increase over the course of treatment might play a role in keeping in check the viral load going further. I might be completely wrong on this, but this is just my intuition on what could have happened.
Great, and I think that's a very sound hypothesis. It's really nice to see this, and this could be the biological mechanism that explains some of the lower risks that we see for viral infections among people living with HIV once they initiate ART.
Exactly. Yeah, and I think this data set that the CFAR biorepository was able to compile just gives us it's one of a kind in that we have information from before they started ART and after, so we can track how these changes actually occurred over the course of treatment. That's
super. Thank you.
Great. So there were two more questions. And so one from Sean Hughes. Are some of the large priority clones specific for HIV antigens?
We did try to find, we didn't find any publicly available, published T-cell receptor clones to be largely dominant in these repertoires, which are HIV specific.
Thanks. And the last question from Daniel Reeves. If I saw this correctly, were the biggest clones in non-suppressed much bigger than in the other suppressed individuals? The y-axis were different, I think. Were those big clones specific for anything, like for example, HIV?
Yeah, so this, if the y-axis here, at least in the top 50 clones, will show the frequency of the clones. And we did find that the two individuals who showed a lack of viral suppression had much more expanded clones, but we did not find any published antigenic specificity of these sequences. While we can posit that they might be HIV specific just because of their prevalence across time, it's hard to actually say that they are.
Okay. Well, thank you very much, Sashi, for a great talk and answering so many questions. Yeah. So
thank you so much
for attending.
Yeah. Thank you.